the idea behind the study is we're dealing with a rare and heterogeneous group of patients who, in the relapse refractory setting, really don't have a standard of care for how to approach their clinical management. And so we essentially compiled a large database that came together into the PETAL Consortium. The centers that contributed data are from all over the world, and each contributed patients to the study, compiling a large data set that allowed us to come to the conclusions that we came to. The question we wanted to ask is, how do sort of novel single agent therapies that have been introduced into the disease space in recent years stack up against sort of conventional chemotherapy regimens that have been around for a long time that come with, associated toxicity and challenges with administration. Furthermore, can we understand which patients are going to derive benefit from these novel single agents, or what type of novel single agents? So, is there a way to prognosticate, who is going to do well with a single agent, and can we make sure to make that drug available to that patient?
We recognize that numerous prognostic risk factors and scoring systems like PIT scores and IPI scores we use in the clinic were available, and there were histological subtype-specific prognostic scores for ENKT cell lymphoma, AITL, and ATLL patients. But we recognize that there's no scoring system that specifically addresses the variability in survival outcomes for relapse and refractory patients at this time. So we aim to design a new scoring system using this one of the largest international global cohort. I could go into specific design of our study, how it's a retrospective global cohort of 925 relapsed and refractory mature T-cell natural killer cell lymphomas from 13 institutions representing 10 countries across the 6 continents. And we believe this is one of the largest retrospective global cohort. And they were diagnosed with lymphoma between 2010 and 2021. And as Leora mentioned, had to receive either cytotoxic or chemotherapy or novel single agents for second-line therapy to be eligible. And for the method section, we first split this global cohort into 80% training and 20% testing set. And we identified that there were 21 available demographic, histological, laboratory, and radiologic non-treatment characteristics we had in the data. And we identified 11 variables on univariable analysis to be associated with inferior overall survival from the start of second-line treatment. And we did the step-by-step selection in the multivariable Cox regression. And with clinical consideration, our final multivariable model included 6 of the 11 variables based on the highest concordance index using the testing set. So the 6 variables in the final model included age greater than 60 and primary refractory disease in contrast to relapse status, and histological subtypes other than AITL, and extranodal sites greater than 1 site, and Ki-67 proliferation index being 40 or greater, and absolute lymphocyte count around diagnosis being lower than lower limit of normal of each site, which led to the 6-point scoring system named the PERT score, which stands for Prognostic Index for Relapsed Refractory Mature T-cell and Natural Killer Cell Lymphomas.
We have this new scoring system with like 6 different score from 0 to score 6 as the maximum number of score that a patient can get. The way we are doing this per scoring system is that we assign a score of 1 to each unfavorable feature mentioned by Angela. So essentially all the clinician needs to do is like take the patient profile and look at all the 6 different features and see if the result matches with the unfavorable feature identified in our scoring system, so that each patient will get from score 0 as the minimum to score 6 as the maximum. And we observe that as the score goes up for the patients, there is a decline trend of overall survival since second-line treatment. And this matches with our, I guess, hypothesis that as the greater the score is, the worse the survival outcome is. And later, the patients are stratified into 3 different final risk groups. So the low risk group has has 0 or 1 risk factor, and the intermediate risks group has 2 to 3 risk factors, and the high risk group has 4 or more risk group factors. And we see similarity of survival between the predicted survival and their actual survival. So later, we also compare our performance of the PERT score with other commonly used scores like PIT score or IPI score, using our test set and also an external validation set. So the way we do this kind of validation is by bootstrapping the testing set 1,000 times, and then take the average to see how the performance goes. And we see that our PERT score has a better prediction probability with average C index of 0.7 compared to the IPI score, which has an average C index of 0.56. And the PIT score has the average C index of 0.59. We also conduct the paired t-tests between IPI and PERT and also PIT versus PERT. And we see that there is a statistical significance of performance difference, which shows that our PERT score really does better than existing scoring systems like PIT and IPI on the testing set, also on the independent set. We also really want to make this like scoring system like available for clinical use. So we developed this web-based calculator specifically for PERT score that can be available to the investigators, the clinicians, and or like patients who are interested in knowing more about this disease. This online calculator is hosted on our PETAL Consortium website. So essentially, we can first go to the website, and then we click the PET score calculator here. And here we have some, I guess, like explanation about what it is about the PET score calculator. And here is the calculator itself. Essentially, we have 6 different risk factors, and they're just like put here as like a question for the user to enter yes or no. So suppose someone is above 60 and is also primary refractory, and also the person is AITL, so that's going to be no here. And then the number of actual nodal involvement is greater than 1, so yes. And then maybe Ki-67 is smaller than 40%, and then maybe the absolute lymphoma count is less than the lower limit of the normal, so that's going to be yes. And then after we answer all the questions, we're going to submit here, and then we're going to have the PERT score as 4 here. And we also see this plots of the predicted survival outcome of our PERT score being 4. And because we have 4 or more risk factors, so we're going to be listed in the high risk group. So this is our kind of predicted survival outcome as being in the high risk group. And also we have some more detailed statistics about our 1-year predicted survival chance, 2-year, 3-year, etc. So and also we have some like disclaimers here for more detailed information about like what data is used to develop this tool, and also what are other relevant information to be known here. I think this is a very easy to use tool, and this will really help the clinicians to have a more comprehensive understanding of the patient survival, especially for the relapsed or refractory patients when they are making clinical decisions in the clinic. So they can basically just go to this calculator and calculate this and see the survival outcome.
We had several centers throughout the United States, Australia, Brazil, South Korea, South Africa, Saudi Arabia, Japan, Italy, as well as India. So we have representation really from all over the world. And I think one question that could come up when extrapolating these conclusions is, is this just a US data set? Is this sort of localized to a certain area and we can't apply this to our patients? And I really think an effort was made to include patients from all over the world so that, the conclusions could be as generalizable as possible. I think one of the major things is that there's a need for consortiums like this in rare diseases to, sort of take a international, multinational foot first and include a lot of different centers in compiling data like this so that we have enough power in our studies to come to meaningful and statistically significant conclusions. So I think that's sort of one, this is sort of one big step forward within the field of T-cell lymphoma. This is a rare and heterogeneous group of diseases, and this collaborative effort, in the PETAL Consortium, really using genomic and molecular data and having really highly annotated information from a diverse group of patients really allows us to ask the important questions that were sometimes a little bit limited in asking in clinical trials and other settings where we have sort of fewer patients enrolled on the study. And then I think, Jessy obviously can talk more specifically about this, but sort of the implementation of machine learning in this context as a way also to draw meaningful conclusions. I think this study kind of frames sort of a new way that questions could be approached and asked within the T-cell lymphoma world outside of clinical trials.
I just wanted to emphasize again that this is a new calculator that's specifically designed for relapsed and refractory patients that we know that has inferior survival outcome than the general T-cell lymphoma patients. And also, as Leora mentioned, since this is the largest global cohort, we were able to do a lot of subgroup analysis that we weren't able to do with a smaller cohort previously. That really takes into account of the heterogeneous nature of the T-cell lymphoma in terms of treatment regimen and also the histological subtypes. Just like other most retrospective studies, one of the limitations of our study was missing data. So we had a total of 925 patients, and we had to exclude 162 patients who were not followed up since second-line therapy, and then 515 were again excluded because we had missing laboratory variables such as Ki67 and absolute lymphocyte count. So which still were, we had enough patients to create this calculator, but we hope to overcome this missing data issue in the prospective cohort study that we plan to launch through the PETAL Consortium that we will soon talk about.
In building sort of this consortium involving, numerous different countries, of course, there's sort of varied treatment practices in the second line setting amongst different academic centers and sort of restricted access to novel single agents in some countries relative to others. And of course, sort of like a lack of, central pathology and radiology review. And I think all of those things may impact study conclusions and our limitations to be acknowledged. But at the same time, I think, again, we've mentioned several times, but these are rare diseases. And, I think this is, in a meaningful effort to draw statistically significant and informative conclusions from this patient population. I think this is something that we have to acknowledge is there, but also still continue with the work of, compiling these data sets to get the best information that we can.
One very important feature about our data set is like the heterogeneity within the patient cohort. We also, I mean, one other challenge is that we don't have the luxury of like having millions of like patients data. So we only have like 925 patients to start with. And so to avoid like problems like overfitting or drawing overfitted conclusions of it over there, we really emphasize on like having this training testing split and also an external validation set to test our scoring system to make sure that it's not just works on within like the training set, which is like fitted perfectly for the model, but also works on like the testing set, also works for external validation set, and so that like the conclusion will be valid like outside like our data set. And of course, we also have this online calculator over there, so that the clinicians can start work to use them, and maybe they will also provide feedback in the future to see like how they feel about the calculator and if they have any more inputs within like new data and other perspectives.
Just to introduce the project that Dr. Mark Soriel, who is one of our leading investigators at the PETAL Consortium, is that he compared the titled forecasting optimal treatment sequences in relapsed and refractory T-cell lymphoma patients, which compare the overall survival since the second-line treatment between subgroups that receive different sequences of treatment of either conventional chemo or single agents such as epigenetic modifiers or small molecule inhibitors. So they have 12 different scenarios in the second and third line treatment, and he identified the more ideal treatment sequences scenarios in specific subgroups, which is more to come.
I also want comment on like the method development within the PETAL Consortium. So essentially, we hope to advance the more like novel machine learning algorithms, like maybe like causal survival analysis methods, to debias the retrospective data, which is often like have some data collection bias or other problem there. So we have this. We hope to have like more advanced methods that can be helpful to really uncover the underlying causal mechanism within the data sets and also have more accurate estimation and prediction from the retrospective data.
I think ultimately the goal is to expand this further, in addition to within the PETAL Consortium, sort of looking at all of these questions and this data prospectively. I think also expanding to additional countries, and there is some interest from additional sites to collaborate on this consortium. So we're looking forward to more growth there. The other component, I think, with the prospective lens is that hopefully we can really incorporate high quality molecular annotation for all these patients. I think that is one thing that will be added in the next phase of this project that was not as well annotated in our retrospective cohort. And then, ultimately with this highly annotated clinical and molecular data from a diverse array of countries with a robust number of patients, we're hopeful that this PETAL Consortium can support a clinical trial network across the world for sort of expedited approval and access to drugs, something that really would benefit these patients and is sort of one of the challenges within the field.
Yeah, just to add a little more detail to what Leora just shared. So we will have exhaustive clinical data in addition to what we had for the retrospective study, would be like quality of life and pathology confirmed by the pathologist panel, and also genomics data, also confirmed by the genomics experts, and incorporation of machine learning. That we believe that incorporating all of these very granular data with that we can move on to precision medicine with use of AI that will allow the allow us to really understand the outcomes and treatment personalization for specific groups to support the clinical decisions in the real world, not just in the U.S. and sites with great resource, but also in the developing countries around the world by sharing our results.
We also want to use like PETAL Consortium as like a platform where researchers from different backgrounds can start to collaborate. So, I mean, people like me more from like the method perspective, like machine learning backgrounds, and also like clinicians, like we all can come here together to work on collaborate and understand the like each other's question or a challenge so that we can together combat this very rare but also dangerous cancer disease.
We just believe that this will just be the beginning of many multinational consortiums fighting for the rare diseases like T-cell lymphoma.